what happens to the size of the lymph node? It gets bigger as we mount an immune response. So that's a proper response to the lymph nodes. Get bigger as we're mounting an immune response. Now, there are two types of immune responses that are possible. One is called a cell-mediated immune response. And that's carried out by T cells. A cell-mediated immune response is a direct attack upon the offending agent, the foreign particle. Okay. So T cells carry out cellular immunity, or cell-mediated immunity. And then you have B cells. When B cells are stimulated by a foreign antigen, they divide and differentiate into plasma cells that in turn rev up and start producing immunoglobulins, which is a fancy word for antibodies. These antibodies will enter into the bloodstream and they will target the same bacteria that entered into the foot. Okay. So they'll travel via the bloodstream and coat those bacteria that are sitting and proliferating within the tissue of the foot. And those immunoglobulins will ultimately lead to the destruction of those bacteria. But this is more of an indirect attack. The B cells themselves are not traveling down here to attack, as would be the case with T cells. Instead, the B cells are producing antibodies that are going to coat the bacteria. And in coating the bacteria, it's going to make those bacteria more easily recognized by other, for instance, phagocytic cells that will then attack them. Okay. This type of immunity, which involves the production of antibodies, is called humoral immunity. Humoral immunity. So you have cell-mediated or cellular immunity and humoral immunity. Now, we said that the lymphatic system consists of a network of vessels, but also tissues and organs. Okay. By definition, a lymphatic organ consists of lymphatic tissue that is surrounded by a capsule. It's encapsulated. This is a connective tissue capsule, like that's connective tissue capsule. Okay. If a tissue isn't fully enveloped in a capsule, then it's not a lymphatic organ. We would simply refer to it as lymphatic tissue. So, for example, our tonsils are not covered completely within a capsule. So our tonsils aren't considered lymphatic organs, but instead we think of them as lymphatic tissue. Right. Scattered throughout our body are little lymphatic follicles, little clusters or collections of lymphocytes. Those lymphatic follicles, wherever they're found, would not be considered organs, but instead lymphatic tissue. Okay. So to be considered an organ, it has to have some type of encapsulation. All right, so what are the lymphatic organs? They are the red bone marrow. Its encapsulation, by the way, is the endosteum. The thymus, lymph nodes, and the spleen. A lymphatic organ is considered a primary lymphatic organ if it's the site where either lymphocytes are produced and or mature. Okay? When lymphocytes mature, we say they become immunocompetent, which means they're capable of mounting an immune response. Okay? So the primary lymphatic organs would be the bone marrow and the thymus. Secondary lymphatic organs are sites where lymphocytes, after they become immunocompetent, travel to perform surveillance. So these secondary lymphatic organs store lymphatic cells. And they'll play a role either in the filtration of lymph in the case of lymph nodes, or the blood in the case of the spleen. You see, our lymph nodes filter lymph. The spleen filters blood. So lymph nodes and spleen are considered secondary lymphatic organs. How would these bacteria get into the bloodstream? Well, if you have such an abundance of bacteria, we're taking bacteria in this case, that are carried via the lymph into the lymph node, such that the capacity of the lymph node is to filter out those bacteria is overwhelmed, then some of these bacteria are going to make it then past the lymph node and into the efferent lymphatic vessel. And once they make it to the efferent lymphatic vessels, they'll be carried into the bloodstream. Okay? If we have an intact spleen, hopefully then, the spleen will filter out those bacteria from the bloodstream and provide protection. If you don't have a spleen, then you're at a much higher risk of sepsis, which is a bloodborne infection. You could get 4+6 because you lose that protective mechanism, that safeguard to protect against bacteria that do in fact enter the blood. So what this slide is demonstrating is that all lymphocytes, whether they're B cells or T cells, are other lymphocytes, because you have other lymphocytes that are not B cells or T cells. Okay? They're all produced in the bone marrow. Okay? B cells are going to go ahead and proceed to become immunocompetent. They're going to mature within the bone marrow. Whereas very, very immature T cells will leave the bone marrow and enter into the thymus. Sometimes we call these pre-T cells. And within the thymus, they will mature and become immunocompetent. So T cells mature within the thymus, whereas B cells mature and become immunocompetent within the bone marrow. Okay? Once they become immunocompetent, they will then travel via the bloodstream to secondary lymphatic organs, like the lymph nodes and the spleen, where they perform surveillance. In one case of lymph, in the other case, surveillance within the blood. Now, they're not going to spend their entire life within that secondary organ. Instead, they may spend some time, weeks or months, before then they enter either into the bloodstream or lymph and travel to another secondary organ. So they can migrate from one secondary organ to another. So, we're going to start today by talking about my friend the thymus. I call him Imus, the thymus. Just because I get bored. When we dissected the dog, some of you saw remnants of the thymus within the superior mediastinum. Okay? You ever see that movie, The Miraculous Case of Benjamin Button?
Yeah.
How Benjamin Button starts off life old, big, and then as he gets older, he gets young and smaller. Kind of a crazy story. But it reminds me of the thymus, because the thymus is largest very early in life. And then as a person matures, as the person advances through puberty and enters into adulthood rather, the thymus gets progressively smaller. In fact, it starts to become infiltrated with fat. It starts to atrophy, and you start to see some scar tissue in its place. So when we dissect a very young dog, sometimes you can see a very, very prominent thymus. But as that dog ages, the thymus gets smaller and smaller and more difficult to identify. It's a bilobed organ. So usually it looks something like this. It's got a glandular. It kind of looks like that. Okay? And it usually sits anterior to the great vessels. So many times when you first open the mediastinum, you don't immediately see the great vessels because it's surrounded by connective tissue, and then the thymus sits in front. Now, like a lot of solid organs within the body, there are two regions, an outer region called the cortex and a deeper region called the medulla. That's common to a lot of solid organs. Well, it turns out that the pre-T cells, the immature T cells that are migrating from the red marrow, occupy the cortex. As it says here, they're in that position, they begin to proliferate and mature. In fact, the stroma of the thymus, the term stroma means the framework or the mattress upon which other cells sit. When we say stroma, we're talking about like supporting actors, not the main characters. Well, the stroma of the thymus consists of a type of cell called an epithelial reticular cell. This is different from other lymphoid organs which have a reticular connective tissue. Instead, it's mostly a cellular stroma, and it's made up of these epithelial reticular cells. Well, these cells secrete hormones called thymocyns that target the pre-T cells, that stimulate their development and maturation. And as these pre-T cells mature and become immunocompetent, they move into the medulla. And we'll see the cortex and medulla in the next slide. Now, the only role of the thymus is that it serves as a school, in a sense, for T cells. The thymus doesn't participate in immunity, in the sense that it's not filtering blood or lymph. It's simply producing T cells. There are no B cells in the thymus, only T cells. You know, it's like going to an all-girls school, high school. There are no guys there. Same thing is true of the thymus. There are no B cells, just T cells. So it's the only organ, the only lymphoid organ that does not directly fight antigens. It's just a school. This is a picture of the thymus under a microscope. It's broken up into lobules. Okay, in other words, it's got, you don't see it well, but it's got this connective tissue capsule, which sends in these little extensions that break up the organ. These extensions, we can even call these trabeculi. Or you can call them septa. S-E-P-T, plural is A-E, septa. Okay, so these are little inward extensions of the capsule that serve to break up the thymus into lobules. But notice, so one of the features of the thymus is it's highly lobulated. It has connective tissue septa that are subdividing the organ into lobules. And then the darker staining tissue here is the cortex, the lighter staining region within these lobules, medulla. In the beginning, when you first start looking at lymphoid organs, you're going to have difficulty differentiating the thymus from the spleen from the lymph node. because all three of these organs are just filled buku with lymphocytes that look like tiny little dots that give the gland its appearance. Okay? But after a while, you're going to start to recognize the differences. Now, one of the features that differentiates the thymus from other lymphoid organs are these light-staining pink structures within the medulla called Hassel's corpuscles. Now, some books will call these thymic corpuscles. What these look like are little light pink-colored swirling structures like this. You may see some nuclear fragments. And they're thought to represent degenerating epithelial reticular cells. So as epithelial reticular cells begin to degenerate, they cluster together to form these Hassel's corpuscles. And so it's a feature that allows differentiation of the thymus from the lymph node and spleen. Okay. Now I'm going to draw a lymph node. A lymph node is a bean-shaped organ. Yeah, bean-shaped. Like this. Except it's not near that big. Some of you, when you dissected the dog, you came across some mediastinal nodes. Okay. They're generally less than 2.5, much less than 2.5 centimeters in maximal diameter. In fact, usually they're less than a centimeter in maximal diameter. So I'm going to draw one of these again, make it a little bit bigger. Now, along the convex surface of the lymph node, let's put it in the capsule. Capsule. This is the capsule. Around again. This is dense connective tissue. And along the convex surface, you're going to have